Good afternoon, everyone. Thank you for joining us for the endocrine grand rounds today. And we are very happy to have Dawn Castor talk today to us about precision medicine in lupus nephritis. Dr. Castor is from Louisville and went to Case Western for her undergraduate studies, but came back to Louisville for medical school, and she has been here since 2002, completing her medical school and residency in internal medicine. She was chief resident, and then did clinical nephrology fellowship, followed by research fellowship in nephrology. And she has risen through the ranks. Now she's an associate professor. She has also been very successful as an investigator and currently has a R01 grant from the NIH, and her interest has been in glomerular disease and particularly with respect to lupus nephritis. And she has been very kind to us and gave us a talk on a similar topic about a year ago. And today we are very happy to have her back and update us on this topic. And welcome to Tom.
Yeah. For those in the virtual space, thank you for being here, and for everyone that made it here in person. I'm so honored to be here today to talk more about precision medicine in lupus nephritis, which is kind of the ongoing subject of my research. And I have some exciting new data to add to last year's talk. So if you were here last year, there will be some review, but there will be some new data to look over. And I'm excited to kind of give a talk about a translational process, you know, of a relatively rare disease. And while it may not seem directly related to endocrinology, there are a lot of lessons to be learned with lupus. And I'm trying to advance my slides. Sorry, that's why I'm.
Put the cursor inside and click in there.
Okay, inside.
Just click there, and now it should.
Oh, okay. Sorry.
Yeah.
I should have. So I have some disclosures. I do some consulting for various pharmaceutical companies. I'm on the Speakers Bureau. I am funded by the NIH, and I'm also the local PI for clinical trials sponsored by Cletus, Travere, and Alexion. None of these affect the topic of discussion. So first, I'd like to identify some current diagnostic and treatment gaps in lupus nephritis, highlight the heterogeneity of disease in lupus nephritis, and then provide examples how precision medicine may be applied to lupus nephritis in the future. So lupus nephritis carries some health disparities and a significant disease burden. Approximately 50% of patients with SLE or systemic lupus develop lupus nephritis. Up to 30% of those patients, especially those with proliferative forms of lupus nephritis, will progress to end-stage kidney disease. And complete remission occurs in less than 50% of patients with standard of care therapies. So we're really not great at treating this disease. So there are significant health disparities in lupus nephritis, and and SLE as a disease disproportionately affects women and minority populations. So 9 out of 10 patients with SLE are women. Patients with African and Hispanic ancestry are more likely to be diagnosed with both systemic lupus and lupus nephritis. And patients with African and Hispanic ancestry are more likely to develop end-stage kidney disease from lupus nephritis. And this is from the CDC website on lupus. But in general, this disease disproportionately affects women and minority populations, and it affects women of childbearing years. Talking to an endocrine audience, we do know that this disease is also hormonally mediated. So these are some cases, not my cases, but you can't come to a lupus talk without me showing a picture of Selena Gomez. So just to highlight, this disease can be silent, and patients can have advanced renal failure without looking very sick. So this is Selena Gomez, Tony Braxton, and Nick Cannon. All three have had lupus affecting their kidneys. Selena Gomez probably the most severe in that she ended up in full-blown kidney failure requiring kidney transplantation. And for those clinicians in the audience, I think it's really important to highlight that these patients may not look like your typical sick patient, but they can be very, very ill without appearing that, because it mostly affects young patients. So I put in my talk that we're going to be talking about precision medicine. So what is precision medicine? So this comes from the NIH. Precision medicine is an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. And lupus is a good disease to kind of tackle this precision medicine approach, and part of that is because there are so many factors that go into the pathogenesis of the disease. The pathogenesis of lupus is very complex, but in this review article by Sokos, he manages to put it all in one beautiful slide. And I really like this slide because it is a multifactorial disease with genetic, environmental, immunoregulatory, hormonal, and epigenetic factors. And in each individual patient, one of these may be playing a larger role than others. And essentially, these factors lead to loss of immune tolerance, the development of autoantibodies, immune complex formation, which triggers cytokine release, T-cell activation, as well as other immune cell activation, and eventually downstream organ damage. It is a systemic disease, so while as a nephrologist I'm really focused on the kidney, this disease can affect not just kidney, but skin, lungs, brain, and heart. So really a multi-system disease. So for lupus nephritis, really it's, again, genetics play a large role, also environment, leading to loss of immune tolerance. There are additional triggers, and that these are generally considered environmental triggers, both hormonal and infectious triggers can lead to this autoimmune response. And essentially, patients will lose immune tolerance, develop autoantibody production, immune complex deposition into the kidney, and organ damage. That organ damage then leads to perpetual cycle. So you have further inflammation in the tissue that leads to this kind of vicious inflammatory process. In nephrology, really we focus on three main classes of lupus nephritis being clinically significant. So the proliferative forms we usually refer to as class 3 and class 4, and then class 5 is membranous. You can have a combination of class 3 and 5 and 4 and 5. Typically, class 1 and 2 are preclinical, and you don't really have severe enough clinical features to warrant a kidney biopsy at that point. And this is just a picture breaking down the different classes. So class 3, focal proliferative, is characterized by subendothelial immune deposits. Most of these patients also have mesangial deposits. And like I said, they can have subepithelial deposits, which are characteristic of class 5 or membranous lupus in combination. The main differences between class 3 and class 4 are that in class 3, less than 50% of the glomeruli are involved. In class 4, greater than 50% are involved. In class 5, you get these deposits under the podocyte, or the specialized epithelial cell of the glomerulus, and often leads to podocyte foot process effacement and proteinuria. So all of these classes are associated with increase in proteinuria. Usually with class 3 and 4, you also get dysmorphic red blood cells, along with that proteinuria. And in class 5, you can actually have nephrotic range proteinuria because of the location of the deposits. So in identifying biomarkers in lupus nephritis, and that's one aspect of my talk, is trying to identify better biomarkers for a more precision medicine approach to the treatment. So what is a biomarker? So I'm using these kind of buzzword terms like precision medicine, biomarkers. A biomarker is something that we use every day in clinical practice. Now, novel biomarkers are always trying to be developed, but essentially it's a characteristic that is objectively measured and evaluated as an indicator of either a normal biologic process, a pathogenic process, or response to therapeutic intervention. So while this is a big buzzword term, we use biomarkers every day. Creatinine is a biomarker. Urine protein is a biomarker. So it's a fancy word for something we've been doing a long time. So we have our traditional lupus biomarkers. So as a tissue biomarker, we have the kidney biopsy, fairly invasive biomarker, if you ask one of my patients. Usually they don't want to go through it a second time, although sometimes I convince them to. Serum creatinine level, proteinuria, which we usually like to quantify because urine concentration can really make a big difference in how much protein looks in a dipstick or simple urinalysis. So we usually use a urine protein to creatinine ratio or a 24-hour urine measurement. Hematuria and urine sediment, we are looking for red blood cell casts, white blood cell casts, any signs of inflammation in the glomerulus. Then you have kind of serologic markers, so anti-nuclear antibodies are characteristic of lupus. Anti-double-stranded DNA antibodies tend to be a little bit more specific and are associated with lupus nephritis, but not perfectly. And then complement levels are often depressed, especially in active proliferative lupus nephritis. However, there are limitations to these traditional biomarkers. So this comes from a study where by Dr. Malvar, who convinced her patients to do protocol kidney biopsies, and basically following patients over the course of their disease, found that there's discordance between traditional biomarkers, so things like proteinuria, hematuria, and kidney function, and disease activity. So here you can see that there are a number of patients. So this is looking at the NIH activity index. So higher means more inflammation on the biopsy, lower means less inflammation on the biopsy. And you can see that there's some persistent histologic disease activity in the kidney of some patients despite clinical remission. So while the vast majority of patients in clinical remission had a low activity index, 29% still had activity indices above 5. So you can see here, there's still a significant number of patients, while they seem to be in clinical remission, that have activity in the tissue, which should really tell us more about what's going on. And then you have patients here in figure 2 that have persistent clinical disease activity. So they have proteinuria over 500 milligrams, despite complete histologic remission. So 62%, and this represents a larger portion, so 62% of patients with complete histologic remission still had persistent clinical activity. And this poses a problem when it comes to treatment of the patients, because over here, you might be, correct in the majority of patients, but in about a third, you might be taking them off of immunosuppression early before the tissue really is showing no inflammation, and putting them at risk. And then over here, on the flip side, you might be exposing the patients to immunosuppression longer than they need to be. So really, we need better biomarkers that can tell us more about disease activity. So the ideal biomarker for lupus nephritis would identify those at risk for developing disease, distinguish between active and chronic disease, guide choice and duration of therapy, and be reproducible in a commercially available assay at a reasonable cost. Okay, I know this is a big ask and we're probably not going to have one. So there's no single biomarker that can do that. It will probably be a combination of genetic, urinary, blood and serum, and tissue biomarkers that really bring us to a new stage of precision medicine in lupus nephritis. So I'm first going to go over genetics because this is basically research that started really at the training level. So I became involved in this project a little over 10 years ago as a fellow. And this is just, again, from that review article by Sokos, looking at genes associated with systemic lupus. And I'm going to focus on this one gene called TNIP1, and I'm going to tell you the story about TNIP1. But essentially, you can see there have been many genes associated with systemic lupus, so this isn't, not by all means, not saying this is the only one or most important one, but it's the one that I've spent studying and trying to understand more about. So this gene primarily involves dendritic cell function and interferon signaling pathways, as well as other innate immune pathways. And even back in 2011, it was identified as one of the many genes that may increase risk of SLE. And then you can see, even dating back to 2009, there were GY studies that identified TNIP1 as associating with SLE in different populations. This is in 2009. And then this comes, this is really the work I did as a fellow. So we identified, and just to be confusing, TNIP1 codes for the protein ABIN1. They don't have the same name. I wish they did. But we did a study basically identifying ABIN1 dysfunction and TNIP1 variants as increasing the risk of lupus nephritis. So this is the genetic data. It came from a collaboration with a consortium called the SLEGEN Consortium, which is why there are so many authors listed, that all contributed cases. And you can see we have a large number of samples from different ancestry: European American, African American, Asian, Gullah, and Hispanic. And this was to identify whether there was a difference between SLE and SLE with nephritis and without nephritis, and of course compared to healthy controls. And we identified two risk variants. This one here is the one that we've gone on to study further, is this RS4958881 variant that was statistically significant to associate with not just lupus, but increased risk of lupus nephritis. In the African American population, we also identified another variant that was significantly increased risk of lupus nephritis in patients of European ancestry. So this is a later review of not just lupus genes, but lupus nephritis genes. And I was really excited to see, this was in 2015, just a couple years after our paper, and TNIP1 is on this list now of not just being associated with lupus, but also increasing the risk of lupus nephritis. And you can see there are multiple different pathways of gene variants that increase the risk of lupus nephritis. So you have immune complex clearance, intrarenal pathogenesis. This includes APOL1, which we know increases the risk of end-stage kidney disease in patients with hypertensive kidney disease as well. Innate immunity, so there are several genes there, and adaptive immunity. And when you're thinking about treatment options, you think, well, maybe targeting innate immunity in someone with this variant versus targeting adaptive immunity in someone with some of these other variants may result in a more precise way of treating our patients. But we don't know that for sure, and we really need to understand more about what these gene variants are doing. So again, TNIP1 codes for the protein ABN1. And what is ABN1? ABN1 is the A20 binding inhibitor of NF-kappa B. And this, I'm not a biochemist, full disclosure, but this demonstrates, so essentially ABN1 in its usual form helps prevent this IKK alpha from going over here and complexing with I kappa beta alpha. Sorry, I'm gonna butcher these names. And basically, when this is bound here, it's in a non-active state. So once I kappa beta alpha comes off of this complex, P65 and P50 are able to translocate to the nucleus. So what is NF-kappa B? I know it's talked about in a lot of disease processes, but essentially it's a transcription factor. So it's only really active when it's in the nucleus, and when P65 and P50 are able to cross over, it stimulates a bunch of cytokine and chemokine responses, which I have here on the next slide. So NF-kappa B regulates a lot of different processes. So you have increased cytokine production, increased inflammatory response, increased chemokines. It affects cell cycle regulators, and of course affects different immune cells. So it's very much a pro-inflammatory transcription factor. So if you don't come away with anything else from the talk, know that ABN1, which is coded, TNIP1 is a gene that regulates or codes for ABN1, and ABN1 downregulates NF-kappa B. So when ABN1 is not functioning, NF-kappa B goes up. And this is kind of, again, our kind of hypothesis of what is going on. So we think that these TNIP variants decrease ABN1 function, which leads to increased ABN1 activity through various pathways. So you get increased autoantibody production, circulating monocytes, and now we think there are probably more innate immune responses that are being regulated, and I'm going to go over that more in this talk. But essentially, we think that this may lead to increased severity and progression in patients with lupus nephritis. So what are some, stop.
Let me just ask a question here.
Yeah.
Now, so would you think that a process that you just talked about was more universal in lupus, or would you think it would be more related, and you talked about personalized medicine to a particular class?
Yeah, so the question is, do I think this is more universal versus for lupus nephritis? So the variant that we've identified, the TNIP variant RS4958881, confers increased risk of not just developing lupus, but developing lupus nephritis in the African American population. So it is a risk allele. And, we certainly NF-kappa B plays a role in several other disease processes, but this is the process we're focused on in lupus nephritis. So, coming forward, so that initial study, we were using consortium data on genetics. But since I joined faculty in 2013, I've really been dedicated to building a biorepository using patient samples. And our biorepository has a large number of patients with lupus nephritis, and we have a sub-study where we've actually been able to do genetic analysis of those patients and identified a subgroup of patients that have this risk allele. And that allows us to not just say, okay, this risk allele is associated with increased risk of developing nephritis, but it allows us to actually do studies to identify, do these patients have a different immune response? Can we look at serum and whole blood markers to see if there are differences? So that brings me to my next section of biomarkers, so looking at serum and whole blood. And essentially, if you're a patient in my clinic, you get asked if you want to join the biorepository, and if you want to do the sub-study, if you have lupus. And we collect serum at every clinic visit that we're collecting clinical samples, and we collect whole blood as well for additional studies on neutrophils and PBMCs. And this is just a look at what kind of tubes we get. So we typically get one red top tube, one yellow top tube. The red top tube is our serum tube. Just to remind you, you have the clot at the bottom, and we bank the serum for future studies. And then the yellow top tube is whole blood. It has some an anticoagulant here that allows us to take the whole blood and separate out these different layers. So we are able to separate out a PBMC layer and a neutrophil layer that we then save for experiments. The PBMC layer we can actually freeze. Neutrophil experiments have to be done same day. So you'll see this in my acknowledgments, but we have a wonderful graduate student, and I have some wonderful help in the lab. Some of these patients might be coming at 3 in the afternoon that help do these experiments when these patients come in. So this is just a serum analysis for 48 NF-kappa B mediated cytokines. We did this because, we know that ABIN1 helps, downregulate NF-kappa B. And we took 31 lupus nephritis patients from our cohort. 74% were African American, 26% were white, and this is pretty much a reflection of the patient population I see. We found that 61% of the African American patients have the TNIP1 variant. 64% were heterozygous, 36% were homozygous. Half of the white patients were heterozygous for TNIP1 and none were homozygous, and we had 6 healthy controls. And this is just looking at lupus nephritis versus controls, and we found that there are definite differences in cytokine expression in patients versus controls, not surprisingly, in a lot of these inflammatory pathways. Oh, and we also found specifically when looking at this variant versus patients without the variant. So the variant here is in red. AA patients have the common allele. And then we have green in control. So you can see differences in interferon gamma, IL-8, IL-16, and MIG. And we thought this was really interesting because it kind of goes along with the hypothesis that ABIN1 is dysregulated in this population. These are primarily interferon-mediated cytokines. And this is just looking at the names. So we have, everything has 2 names. So it came up as MIG here, but it's CXC9, which affects, and these are just some of the different cell processes. So it can affect neutrophil migration, inflammatory response, granulocyte chemotaxis, and then these are some of the other responses. I won't go ad nauseum, but clearly it does seem like there are differences in the activity of these different cytokines. So this is another study that we did looking at cytoph analysis. So we used actually the PBMCs, not the serum. And if this is a core lab by Yan and Chesney, And Hong Lee was kind enough to do these experiments and also helped interpret these. So research doesn't occur in a vacuum, and we were really thankful to be able to collaborate with their group in doing this analysis. So we took 15 samples. We were debating how many. So they said they could do 15, and we're like, how many controls do we do? We wanted to do as many lupus nephritis samples as we could, and we thought, based on our other data, usually you can see a really big difference between lupus patient and controls. So that's why we have so little controls on this. And this essentially shows you the subpopulation and the heat map. And these are some of the specific clusters that were altered in the lupus nephritis patients compared to healthy controls. And you can see here, this is a CD8 positive T cell subset, which was higher in lupus nephritis and lower in controls. And interestingly, the CD4 positive T cell subset was actually lower in the lupus nephritis versus controls. And then this is a myeloid cell subset, which includes monocytes, dendritic cells, and potentially neutrophils as well. And you might be wondering, why are there neutrophils in the PBMC fraction? There are actually these specialized neutrophils called low-density neutrophils that do show up in that fraction that are actually really important to the pathogenesis of lupus nephritis, because these low-density neutrophils are more likely to net. And I can't get into too much detail on that, but essentially netting neutrophils has been proposed as a big component in the pathogenesis of both lupus and lupus nephritis. So we thought this was really interesting. We showed some clear differences in these groups, and we showed that measurements of serum levels of interferon gamma and associated chemokines may be considered as diagnostic markers for patients with this TNIP1 variant. And then CyTOF analysis demonstrates differences in immune cells in both the lupus nephritis patients that have the variant and all together compared to healthy controls. And that brings me kind of to the next topic, and hopefully I didn't put too much information in here, but so our lab is also interested not just in this TNIP1 variant, but we're interested as a whole in innate immune responses. And some of this work actually made us think, what is the role of neutrophil participation in lupus nephritis, both in patients with the variant and without. And neutrophils, as many of you know, are one of the most abundant immune cells. They're often overlooked because they're kind of the first responders and they die off very quickly. But the role in lupus nephritis is becoming more and more understood, and hopefully not as overlooked as it once was. And through processes including degranulation and phagocytosis and netosis, neutrophils basically get their work done. So their main role is basically as their first response against bacteria or pathogens, and they can attack those pathogens through degranulation processes or phagocytosis. And netosis is another unique way that they can target bacteria. These are the different types of granules that neutrophils secrete in their degranulation. And we have this hypothesis, and this is work that was done a few years ago, that neutrophil degranulation can contribute to injury in glomerular nephritis and lupus nephritis. And this is work that was done on an animal model of immune complex glomerulonephritis. So it's called the anti-GBM model, also can be called nephrotoxic serum model. But essentially, it is a model of kind of a rapid glomerulonephritis. So these mice, get disease very quickly, and develop proteinuria very quickly. So this is some of that work where we demonstrated that animals given this nephrotoxic serum, essentially anti-GBM, develop high levels of proteinuria at 24 hours. Like I said, it's a very rapid model. However, if we blocked neutrophil granulation or degranulation with this TAT-SNAP23, we were able to reduce that proteinuria at 24 hours. And this is just looking at WT1, which is a podocyte marker expression in the two different groups, showing that you have more podocyte damage in mice. Sorry, I'm so used to talking about patients. In mice that were treated with NTS and not given the neutrophil degranulation blocker. So essentially, this demonstrates that neutrophil degranulation, plays a role in immune complex glomerulonephritis, at least in this model. So, we have, of course, mice and are not humans. And this is a big part of our work, is that we really, really want to be using human samples. So we hypothesized that degranulation is different in patients with lupus nephritis. And this is just work showing that in unstimulated conditions, patients with lupus nephritis have higher levels of degranulation. And this is work looking at neutrophils taken basically the day that they come into clinic versus control neutrophils. We also conversely found that if you take normal neutrophils, so we're able to collect normal neutrophils from healthy donors and isolate these neutrophils, and we added pooled serum from patients with lupus nephritis and found that if you add pooled serum to normal neutrophils, there are endogenous activators of neutrophil degranulation in that serum. So this is untreated. And again, these are normal neutrophils. This is FMLF, which we expect to stimulate neutrophil degranulation. And this is looking at the addition of pooled lupus serum versus pooled normal serum. And you see here with CD66B, clearly the pooled lupus serum activated these neutrophils. CD35 also showed a difference, but wasn't as strong here with showing the difference between the pooled. And we're increasing the ends on this. So, and this is looking at an individual example where there was a significant difference. So we had to use quite a lot of serum, and I was afraid to. All my human samples are precious, so I was afraid to use too much at once until we saw an effect. So we started with the pooled, and now we've started to look at individuals to see if they have differences. And you can see here in this individual, CD66B versus CD35, both markers of degranulation are enhanced in lupus serum versus normal serum. And again, this is looking at normal neutrophils. So then another study that we decided to look at was looking at analysis of these granule and net proteins, so looking at these individual proteins in patient serum and urine to see if these were potential biomarkers of disease activity. And this was a custom array that we developed, and we started out with 10 active lupus nephritis serum samples versus 8 controls, and we decided to do active disease because we really wanted to identify markers of disease activity. So this is looking at patients that had over 500 milligrams of proteinuria, and most of them were well above that. these are the different markers we looked at. And we were kind of disappointed when we got the serum because neutrophil elastase was the only one that was elevated versus the control. But then we also looked at urine, and something to consider when looking at any nephritis biomarkers is that urine may be a better reflection of what's going on at the tissue level, because if you have all those neutrophils activated, in the glomerulus in the tissue, you would expect some of those markers to kind of spill over into the urine. So we found a large number of proteins, granule proteins, that were increased in the urine of patients with active lupus nephritis. So again, a bunch of potential biomarkers identifying patients with active disease, and these are the different ones here. And this is just looking at what type of granule type they're coming from. So neutrophil elastase is eosinophilic, lactoferrin, specific granule, MMP9, gelatinase, but there's a large number that we're looking at the specific granules. And then this one, PAD4, which induces net formation. So we found this very interesting. And then we also wanted to look at tissue. And this is kind of early studies that we're looking at. This is the real fancy microscopy that has become available that allows you to look at basically it's almost like tissue cytoph, but not quite, where you're able to look at multiple markers at once. So this is looking at neutrophil markers in a patient. This is a biopsy from a patient with lupus nephritis, with proliferative lupus nephritis, versus control tissue. And you can see a lot more lighting up here, suggesting a lot more neutrophil activity in the biopsy of an LN patient. So the conclusions there are that measurements of urine levels of neutrophil granule proteins could be considered a diagnostic marker in lupus nephritis. Enhanced urinary granule protein levels could indicate renal inflammatory activity in lupus nephritis. Of course, this all needs to be validated and done with more cases. And finally, that targeting neutrophil degranulation and net production may be considered a treatment for lupus nephritis in the future. This is just an idea of some potential future applications. This came from Monroe and James's review of genetic variants in lupus nephritis, but just looking at how potential genes may regulate these different inflammatory processes, and give us a more targeted look at potential therapies in patients with lupus nephritis. So, my hope would be in the future, we'd have a panel of biomarkers. So we might have, patients that are genotyped, and they might have multiple gene variants. But for example, if they had, upregulation of, or if they had the variant that we're looking at in TNIP1, and they showed upregulation of interferon, that maybe a therapy that targets interferon would be a first choice of therapy. In the lupus nephritis world, we're really excited because we've had, a couple new therapies that have been FDA approved in the last couple of years. However, as more therapies come into play, then we have to decide, which therapy is best to start with in each individual patient. And right now, it's just a guessing game. So we start with standard of care, and we might do an add-on therapy that might be B-cell, or, for example, interferon. The first interferon therapy just became approved or anti-interferon therapy became approved in systemic lupus and is being studied in lupus nephritis. But right now we don't really have a way of knowing in an individual patient which pathway is more upregulated. And hopefully in the next decade, we'll get to a more precision medicine approach, kind of like what they're doing with, cancer and chemotherapy. So, final thoughts. Current biomarkers are inadequate to identify disease activity in lupus nephritis. Unfortunately, the standard of care therapies result in complete remission less than 50% of the time. And novel biomarkers have the potential to help with both diagnosis and treatment to improve outcomes in patients with lupus nephritis. And I'd just like to acknowledge and thank all the lupus nephritis patients of mine that contributed to the samples in the study and ongoing studies. We would not be able to do this without them. And honestly, I think it really puts UofL in a unique position that we're able to get, whole blood from patients, almost on a weekly basis and do these experiments. I didn't put this on here, but we also get samples and neutrophil from healthy donors, which if there are flyers around this building, so some people, may have donated, and we want to, thank all of our healthy donors that provide samples. Obviously, research doesn't happen in a box. It takes a lot of collaboration from a lot of different people, and I wouldn't be here today if it weren't for my collaborator, David Powell. Michaela Brady is one of our doctoral students that works in the lab. She's finishing up her PhD. She's been wonderful. She's done a lot of these experiments, stayed late on Tuesday nights when I have my clinic patients coming in. Shweta Tandon is our technician. And these are several nephrology faculty members that have helped contribute to my career and development, and then some of the outside collaborators. And with that, I will take any questions. Here's the event code. I'll leave that on for those who need it. But again, I really appreciate you letting me give this talk. I know it was a little outside of endocrine, but hopefully some of the topics are applicable. I think anytime we're having these new therapies, a big question is always, so which therapy do I choose for my patient? But I'd like to start with the best possible one first before going through several. So I think I'm on time. Yay.
So your controls were all healthy controls?
Yes.
So are there lupus patients without nephritis?
That's a great question. So the question is, do we have controls of lupus without nephritis? And one of the caveats to doing this study is that we haven't had rheumatology here in a few years, so we haven't had a source of control samples, but we are collaborating and trying to get some from Oklahoma and likely from Cincinnati to have some lupus without nephritis controls. I completely understand it is easier to detect difference when you're comparing normals to patients with lupus. And we do plan on, incorporating those studies. We have, and I didn't show this data, but we have actually used patients as a control. So we've used active disease versus remission and demonstrated some differences for some of the biomarkers we've looked at. So that's one great thing about having a biorepository that's, we have clinical data. We can actually use patients as their own controls when they go into remission to see if something's a good active disease biomarker. But we are planning on doing more studies with lupus controls.
Follow that.
Yeah.
Does it go up and down?
Yeah. So we've seen so patients are followed. So now we've been doing this biorepository for almost a decade, actually at least a decade. But so patients with lupus have periods of remission and relapse. And some never go into remission, but the ones that go into remission, we actually have some that, have had response to therapy, were in remission for a period of time, relapse for a period of time. That's one really big benefit to doing this longitudinally. So at least for serum and urine, we're able to look at, in the patients that have a remission and active disease period, we're able to look at 2 different time points. But it's human data. It's not perfect. It's not going to ever be as controlled as animal models. But in animals, they're almost always using a normal as a comparator. Yes.
Interesting talk. And
Yeah.
that's a really good question, I think, that you asked, Doctor. Because, like when there's more proteins in the urine, whenever they were, versus what's in the serum, could that be more due, let's say, increased permeability rather than due to intrinsic differences in mechanism?
So that's a great question, and we actually have disease control. So the question is, when you have patients that have proteinuria and you're finding increased levels of these markers in the urine, is it just because of the proteinuria? So we actually, and this is something that we can do, but we've done it for other biomarkers. I didn't present that data, where we've looked at specific markers in patients, not just with lupus nephritis, but also focal segmental glomerular sclerosis and other proteinuric kidney diseases, to show that it was very specific for the lupus. So certainly it's something that we should consider doing in this, but we have what we call disease controls that help. And then all the urine studies, just to clarify, they were also controlled for urine creatinine level. So anytime you're doing a urine biomarker, it's pretty standard because you can have such variation in urine concentration, to control for urine creatinine to make that more uniform. But great question.
Since you had these differences between African American and British, so what do you find in these?
Yeah, so unfortunately, so we can't break it down. So the question is, we're seeing differences in African Americans versus Caucasian patients. Have we broken down for the biomarkers? We don't have enough N's right now to really get to that. And that's, even in clinical trials where they've enrolled a lot of patients, when you do this subgroup analyses, it's hard because, you're making your N smaller. So sometimes, yeah. So certainly as we increase the numbers, looking at racial differences it will be important. And our population, so we would like to expand. So our population, like I said, is almost like 3/4 African American. We are collaborating with Oklahoma Medical Research Foundation, where their population is much different. And hopefully we'll get samples from them and we'll be able to analyze to see if there are differences. And they're also doing the genotyping too. So great questions.
The TNIP one that you have, so it looks to me like it is something that or whatever not indicates an increased risk for
Yes. Yeah, increased susceptibility.
Is it there in other inflammatories?
Yes, actually, that's a great question. So has TNIP1 been associated with other inflammatory diseases? And it has. So I'd have to look at the list, but it has been associated with other autoimmune diseases, other variants. So yeah, NF-kappa B, like I said, is involved in multiple processes. So so it is not a variant that's just, specific for lupus nephritis. Or it's not let me backtrack. It's not a gene. It's just specific for lupus nephritis. The variant that we're discussing is associated with lupus nephritis. I don't know if that variant's really been studied in other diseases, but other variants within TNIP have been looked at in other autoimmune diseases.
Are there any preclinical specific target?
So, I mean, even steroids affect NF-kappa B. So it's there are some potential that are more targeted towards those pathways, but you know,
So no,
yeah.
for gamma
Mhm.
or it is
Yeah,
for the other side.
yeah. So no, that's a great question. See if
Do you have any questions that
Uh,
you're sharing?
oh, let me look. It looks like that's just the CME code. Is that the chat? Should I see questions?
Correct.
Okay. Okay. Anyone has questions, feel free, or you can email me, track me down. I'm easy to find. But yeah, I think there's a lot more to do, which is exciting because I still have a lot more career
I
left.
mean, obviously, this is something that we need multi-center training.
Yeah.
They don't get much. And some sort of AI program that will
Mhm.
put together the blood and the
Yeah.
urine and all these things together and try to come up with just calculate
Yeah.
something like that.
Our cohort, though, going back 10 years, we have over 50 LN patients. So definitely more than the average nephrology center. But so we do get quite a few. But yes, it will be helpful to, and that's why we're collaborating with other places. And also to get those lupus controls or non-nephritis lupus, which it's always hard though, because they're all at risk for developing lupus. So you always wonder, but it is important to look
For
at
these
those.
measures, they have to be without on.
No. All patients get standard of care therapy. So the question is, you know, for these measurements, are they without steroids? And the reality is when we're doing translational work is we do have the data, and I can tell you that the vast majority of patients are on mycophenolate mofetil, are on some level of prednisone. I didn't show this, but we do have that data. Most patients are on hydroxychloroquine. They're mostly on similar immunosuppressive regimens. There is some variability, but all patients are on standard of care. You can't withhold therapy from patients.
So that doesn't affect anything.
It may have an effect, but we're still seeing differences. Yeah, so an animal model, obviously, it's easy to test, but yeah, in human disease, it gets a lot trickier, but we still see big differences, even though all these patients are on some level of immunosuppression. So even despite that, they're...
How good is the animal model?
So the animal model, so if I go back, here, I'm gonna... If I X this, well, okay. So, well, I'm trying to go back to our TNIP1. So the difference is we know that the animal model, it's actually a knock-in, it's not a knockout, and it affects the polyubiquitin binding of ABM1, so it's not able to bind here. And we know functionally those mice have impaired ABM1. Now, in the human disease, and I should have added that slide back. The slide I deleted because I was worried I was going over. Depending on the variant, the one that we're studying is actually in the non-coding region. So again, it's really hard to know exactly what it's doing.
So the animal with this, okay.
Yes.
Histology look like.
They get proliferative lupus nephritis. And I left out those slides too, but that was part of that paper. So they get proliferative lupus nephritis, very classic immune complex deposition. Yeah, some of the early studies that I did as a research fellow was, doing the staining, the immunofluorescent staining on this mice tissue. So yeah, they develop lupus nephritis. And they develop higher levels of ANA, anti-double-stranded DNA. So they basically develop lupus-like autoimmunity. Yes.
This is, a really good area for personalized medicine because the treatments are not trivial. They have a lot of bad effects. Okay? And it's not like hypothyroidism where you give a person some thyroid and then they pretty much function normally and very clear what the mechanism.
Yes.
Here, some patients respond to certain treatments and other patients don't respond to those treatments at all.
And then you're exposing
There's
them.
a whole
Yeah.
range of bodies, even though nobody really knows if the antibodies are important,
Yeah.
and cellular process, the immune cellular process going on and all of these things. So that even if you can just tease out 1% of people that respond to one kind of medication, and something really reliable.
I appreciate that comment. So, you know, that makes me think I'm in the right field, but no, I mean, it's true. The reason I went in to study lupus nephritis specifically is as a first-year fellow, I was in my second month and I had to put an 18-year-old on dialysis from lupus nephritis. And I was so frustrated because we did cyclophosphamide and we did steroids and we did everything. And she was in such a state of inflammation, there was just no turning back. And I was frustrated. I was like, this is a horrible disease if an 18-year-old's having to start dialysis. And then, you know, I remember Dr. Powell was presenting. I had no lab experience. I had only done a clinical chart review as a resident. I knew I was interested in nephrology, but I was afraid of the lab, so I did a chart review. And then Dr. Powell presented his work on this animal model, and I was like, you know what, I'm going to get my feet wet, do some lab work, and it led to one thing, led to another, and you know, hopefully we're making progress in the disease, but you're right, the treatments are toxic. They're not, you don't want to give someone cyclophosphamide if they're not going to respond, you know? So identifying a more targeted therapy will be helpful. And a lot of these new therapies that are coming out, so we now have 2 new therapies that were just approved in the last couple of years. They target different pathways. One is a calcineurin inhibitor, the other targets BAF or BLIS. Why do cytokines all have like multiple names? I don't know. So, you know, and then down the road, there are complement inhibitors that are being studied, interferon receptor antagonists that are being studied. So identifying who would benefit from which therapy would be really helpful. But we're not there yet. But maybe in my career we'll get there.
I remember it must have been 50 years ago. I followed the Mass General, and they gave I think his name was Cohen. It was either Cohen or Watts, but it was a Jewish name. and there were actually Cohen and Schwartz at the time that were experts in lupus in the Boston area. And he had a patient that he had on corticosteroids, doing very, very well. And then he took her off because, you know, the side effects and everything. And she started having a bad case of lupus nephritis, and he put her back on, and it didn't help, and she died.
Yeah.
And, you know, he said he would never completely take the patient off of corticosteroids if they were responding. Of course, much less was known back then, years ago, than today.
Yeah.
But indeed, it hasn't changed that much.
It hasn't. You're absolutely right. It hasn't changed that much, and I can still remember, because we always wonder, you know, how long do we keep them on the MMF? Because, you know, if they're doing well, you're afraid to, you know, take them off. And, you know, that study looking at biopsies highlighted that some people still have inflammation even when their clinical markers are okay. But we've all seen cases where patients took themselves off and then relapse. But it's really hard to know. That's why all of the guidelines, so we have all these guidelines that say, this is the induction therapy, this is your initial therapy, and then maintenance therapy should last at least 3 years, but we don't know how long, because everyone's afraid to take people off because they can flare. There have been cases of patients that are in complete remission, you take them off, they flare, or even develop antiphospholipid antibody syndrome or, you know, some other disease process that's related to lupus. So challenging.
Lifespan of these, because I,
Yeah.
when I was in Rush, I trained
Yeah.
there.
Oh,
Every other patient I saw,
yeah.
or young women, just
Yeah.
like you said, young women, either Hispanic or African American, or already on dialysis or getting to
Yeah.
dialysis or on the transplant list.
So
I
the,
did
yeah.
nephrology with them. That's where Susan Ho,
Oh,
sort of donated her own kidney to her patient,
yeah.
a pregnant Hispanic woman who was out in Haiti. So, what is because I'm thinking, am I seeing like lupus patients in their 70s or 80s, and I can't
Yeah.
think of many.
So the mortality has improved with new therapies. However, in certain parts of the country, like the Southeast United States, looking at like USRDS data, there's still a really high rate of patients going on to end-stage kidney disease. And we don't really think about long-term effects. So I had this great case, in clinic last week where I had a patient that was in her mid-40s. She had just had another MI, and she was diagnosed with lupus. I don't even know if she had lupus nephritis officially diagnosed, but diagnosed with lupus at age 12. And she has chronic kidney disease now. And her first heart attack was at 37. So the increased cardiovascular risk, the mortality is definitely higher than the general population. It's higher in patients that have nephritis than patients with lupus without nephritis. And downstream from the early nephritis, you have cardiovascular complications.
Transplant.
Yeah, and then transplant-related complications. But even the patients that don't end up with end-stage kidney disease, they have CKD by middle age, they have increased cardiovascular risk. And I hate those, I shouldn't say I hate them, but the cardiovascular risk assessment doesn't include proteinuric kidney disease. It says on there like an asterisk, like some people put it as a diabetic equivalent, but I've had patients with proteinuric kidney disease that are told, oh, your risk score is not high enough to put you on a statin. And then I get really upset because I'm like, okay, this person has CKD3, they have proteinuria, they should be diabetic equivalent. But people don't realize that on those calculators. So brings up a good point, a good teaching point. Like, we need to manage the cardiovascular risk in these patients aggressively because they're very high risk. Especially as they get older. It's hard to think about it when someone's in their 20s. Yeah, So because I had a patient that was like 45-year-old female with IgA nephropathy, had hypertension, CKD3, proteinuria, and she was having some myalgias, and they're like, oh, her risk assessment shows she doesn't really need to be on a statin. I'm like, what? So obviously I'm in rare diseases, so I don't get to use algorithms very often. Just a very good point. These patients end up with a much higher mortality rate. It's improved since the 70s, but not as much as we would have hoped.
So much.
No problem. Take care.